Hello everyone, welcome back to new video. So today is the class 4th in the series of MHSRV Government of Telangana Lab Technician Vacancy Preparation. In this video we will be discussing about Unit 4 which is related to cytology and general pathology. Okay, so these are the topic we will cover in this video. So let's get started with the video. Now, before we begin into really tough details, we will discuss about basics of cytology. So, what is the cytology? Cytology is the study of cells. More specifically, if I will say, to evaluate individual cell or cluster of cells to diagnose certain diseases, including some forms of cancers. ठीक है? तो we are using cells or clusters of cells to make certain diagnosis. So this entire thing is known as cytopathology or cytology. So in the field of cytology, we will be collecting various kinds of samples. So there are cytology sampling techniques. Majorly, it is exfoliative cytology, FNSE, and body fluids. We will discuss about these techniques in detail. Moving forward, now first begin with the exfoliative cytology. So term exfoliative means study of cells that have been shed or removed from the epithelial lining or epithelial surface. तो बेसिकली कोई भी ल्यूमन है, कोई भी लाइनिंग है, उसकी जो एपिथेलियल लेयर है वो शेड होती रहती है. सो जस्ट लाइक ट्रीज इन ऑटम सीजन डू शेड ऑफ देयर leaves, old leaves है ना. Similarly our body cavities or organs also shed some cells. ठीक है, तो we are using those cell in exfoliative cytology. ठीक है. Now there are two kind of exfoliation. One is mechanical exfoliation and other is spontaneous exfoliation. So spontaneous exfoliation means cells are shedding spontaneously है ना, that is without any outer intervention. Okay, so this includes voided urine है ना. You see cells in certain cells in urine है ना. Do we mechanically exfoliate those cells? No, it is example of spontaneous exfoliation. Okay, now we will be talking about mechanical exfoliation है ना. It is in this technique cells are scraped from the surface by using some material, some spatula. Okay, we will scrap the cell from the surface है ना. This is called for example in cervical cytology or pap smear है ना. This is also known as abrasive cytology because we are basically using some instrument to mechanically exfoliate the cells from the lining of lumen. Okay, now in exfoliative cytology there will be two kinds of samples majorly, a gynecological sample and non-gynecological sample. Okay, in gynecological samples there are vaginal smears, cervical smears related to basically female genital tract, which helps in the screening of cervical cancer and hormonal assessment. Okay, common technique is pap smear technique. Okay, and the second kinds of sample in exfoliative cytology is non-gynecology samples, which includes bowel, urine and other fluids. Well is bronchoalveolar lavage. Okay, alveolar lavage. So these two kinds of sample basically we deal in exfoliative cytology. Now the question you will get in exam is basically will come from here in from the pap smear. Okay, pap smear basically means Papanicolo smear है ना. Pap is came from Papanicolo, which is a scientist who do lots of things in the field of exfoliative cytology. Okay, so it is, so pap smear technique has basically conventional pap smear technique. In this we using a iron spatula, looks like this, and we will insert into vaginal canal of female and we will collect sample from these cervix area है ना, by rotation of this spatula, and we will just paint it over a slide. Okay, just paint it, random painting, no certain clockwise pattern, just random painting from the spatula, and then we will fix it and stain it later. Okay, this is called conventional pap smear technique. In this we will be using iron spatula. Now there are certain modern or advanced technique which is comparatively better instead of pap smear, conventional pap smear technique. Okay, this is called liquid based cytology or LBC. In this thing we will using cervix brush, looks like this है ना, this like. So in this also we will just put it into vaginal canal or cervical canal, and we will collect from sample, okay, from this portion, and we will collect cervical sample by the help of cervix brush, just like this है ना. This is also rotated inside 5 to 10 times to collect certain quantity of sample. Okay, then we will just do some techniques. I will tell you about these technique, and we will get slide, clear slide like this, not just like conventional pap smear where you just painted and stained, है ना. This is beautifully organized basically. Okay, this is LBC. I will share two techniques about the LBC. Okay, so there is two types of LBC. One is sure path and other is thin prep. I will briefly explain about it. In this, the important points mainly I will discuss. Okay, so first of this kind of container is came for sure path. Okay, and in this there is ethanol based preservative. Okay, and when we collect sample in cervix brush like I showed you earlier, and we will just drop the brush into the container and send it to the laboratory. In laboratory we will just layer it over a density gradient medium, है ना, which will separate the contents of the vial into depending on their specific gravity. So the diagnostic cell will came under the bottom, and we will just discard the upper portion. Okay, discard the upper portion and resuspend this pellet into an isotonic solution, decant it into the settling chamber. This kind of chamber is called settling chamber, where you have here is positive charge slide which makes better bond with cells. Okay, and here is this thing, this pellet, resuspended pellet na. So slowly under the gravity sedimentation occur and cell will stick with positive charge slide. So this technique is called sure path. In this there is two terminology you have to remember. Density gradient centrifugation we will do, and also gravity sedimentation we will do. Okay, gravity sedimentation, not some artificial sedimentation. Okay, now we will moving forward about second technique, which is called thin prep. So in this technique we will not cut the brush from here or drop the brush from here. We just mix it properly with the solution, methanol based preservative. Okay, now we will just vortex it properly so the cell will disperse into the solution. So, and now there is some methods of cell collection. So there is negative suction, here is the filter paper, and here is the solution, this solution. Okay, है ना. So there is negative suction, so cell will pulled upward and this will intact into the filter paper. Okay, and now there is method of cell transfer. After that, है ना, we will just pushing it with certain positive pressure so that cell will fall over the positive charge slide on the glass side, है ना. This is circular, so we will get the smear like circular, which is shown over here just like this. So I will just tell you what a conventional pap smear looks like. This is messy thing, है ना. This is sure path. We are just having small circle because we are very sure to get a diagnosis from it, because we also drop the brush into it. Okay, and also here is large circle which is of thin prep technique, है ना. So just you have to remember in LBC there is two techniques, sure path and thin prep. Now we will be discussing second kinds of sample in exfoliative cytology. It is called buccal swab or smear. Okay, a buccal smear is a test where cell is taken from cheek inside of mouth. Cells are collected by scraping the cheek from the cotton swab and suspended in water. Then after from that swab we make just a smear. Okay, and we stain the smear with drop of methylene blue and observe under 40x, है ना, and we will look for squamous epithelial cells. ठीक है. तो buccal smear, what is the use of buccal smear? In 50% of cases, confirm the patient sex by evaluating for the presence of Barr bodies. I will tell you what is Barr body. The number of Barr bodies give an insight about the abnormal karyotype of individual. What is karyotype? Karyotype is chromosomal property or arrangement. Basically, we are studying chromosome. So I will briefly tell you about the Barr bodies. Okay. Suppose there is male, female. Male has XY chromosome and female has XX chromosome. Okay. So in more than 50 to 60% of female, there is chromosomal inactivation, है ना, which condense the chromatin, basically which condense the extra X chromosome. Okay, one X responsible for female and other X will be condensed. Okay, looks like this, है ना, ठीक है. So suppose there is XX female, okay, that is normal female na, which has one active X chromosome and other X is inactive. Okay, this is Barr body. Now we have XY male, which is normal male. So X has one X which is active, okay, other is Y. So there is no Barr body in male. No Barr body in male. Okay. Similarly, in case of Klinefelter syndrome, okay, which has XXY, which has basically one extra chromosome. So there is X, which is active as, there is Y, which is active Y, that's why it is male, and there is extra X, which is inactive. So this is Barr body. Suppose there is pattern where 3X, triple X female is there, okay. So one is active, other two is Barr bodies. Basically inactive. So basically inactive X chromosome is Barr body, which we'll see like this in cells when there is a condensation of sex chromatin. Okay, so there is second sampling technique in the cytology is FNAC, which is fine needle aspiration cytology. It is also known as aspiration cytology in whole. Okay, FNAC is a study of cellular sample obtained from through a fine needle, that is very thin needle, under a negative pressure. There is suction which pull the cell into the syringe for diagnostic purpose. So this is FNAC. Okay, so there is certain requirement about the sampling technique, which is called FNAC. So we will have to have a syringe of 22 to 25 or 26 gauge needle, and with syringe attached usually is common, 10 ml is common. Okay, there is a thing called Coplin jar, which has 95% ethyl alcohol fixative, which is recommended. Okay, this is syringe 10 ml. This is piston to pull syringe. Okay, needle also here, forceps and glass slides. Okay. These are the things we basically need in case of FNAC. So let's see a brief about the process of FNAC. We will just clean the site. We will palpate this thing, any kind of swelling. We will just pulling the piston to collect certain diagnostic material, and we will push it on the slide and we will fix the slide. This is basic FNAC technique. Okay. There is some scientists who discovered a technique called French technique, aspiration using a fine needle without the negative pressure or syringe. This technique is known as French technique. Basically we don't need this thing. This is less traumatic to patient and cause lesser bleeding according to those scientists. So this is called French technique. So there are guided versions of FNAC. Ultrasound guided FNAC, CT scan guided FNACs. Now we will make the smear from the FNAC. We will take in the diagnostic material into syringe and needle. We will just push it on the this thing, slides, over a slide, and fix it. Okay, there are usually two slide made after an FNAC. One is for a pap stain, okay, other is for which MGG stain, other is for certain special stain like ZN stain in case of tuberculosis suspectans. Okay, there are two kind of fixation. One is immediately wet fixed into 95% ethyl alcohol or ethanol in Coplin jar. for 15 minutes before the we actually do the pap stain. So in the cytology term, fixation refers to preservation of cytoplasmic and nuclear details or morphology of the cells. Rapid fixation in alcohol, which is called bed fixation as explained above, is essential for pap staining, which brings out the nuclear details clearly, allowing better identification of malignant cell. Okay, and we just allow to dry out the smear like we do for blood smears. This is done for Romanovsky stains like MGG, GIMSA, etc. Okay, so let's just discuss in more detail about the fixation of cytology specimen. Okay, so there should be rapid fixation of smear is necessary to preserve cytological details of cell spread on the glass slide. There is certain cytological fixative we will be discussing here. First is which is used in routine labs or routine fixative. Okay, the process of submerging freshly prepared smear immediately into liquid fixative is called wet fixation as explained earlier. This is the ideal method for fixing all gynecological and non-gynecological smears. Okay, majorly wet fixation is ideal in case of cytology. Okay, the most common and in today's life ideal fixative is 95% ethanol or ethyl alcohol, which we will be using in our labs. So there is old but very excellent fixative also, which is originally recommended by Papanicolo, which is a scientist who has done lot of work in field of exfoliative cytology. It consists of equal parts of ether and also 95% of ethyl alcohol. Two components you have to remember, ether and alcohol. It is an excellent fixative, but ether is not used in most of the laboratory because of safety hazards, odor and hygroscopic nature. It is also highly inflammable. Okay, and many other thing which makes it unrelevant in today's world. Okay, 100% methanol is an acceptable substitute for 95% ethanol, है ना. Methanol produces less sinkage than ethanol, but it is more expensive than an ethanol. Okay, there is certain denatured alcohol unsuitable for human consumption, so there were the some people who just drinks the alcohol from the laboratory, so that's why we made it something called denatured alcohol, in which the major component is same 95% ethanol, 90 parts. 5 parts we will take from 100% methanol and 5 part we will take from isopropanol. Okay, so this is the composition of a denatured alcohol. Now there is a second line of fixative which is called coating fixative. Okay, coating fixative or spray kind of fixative are substitutes for wet fixatives. They are either aerosol applied by spraying on the cellular component or samples, which is dropped on the slide. So basically there is slide and there is a spray, okay, which will be sprayed over the slide, which has diagnostic material or our sample. There are composed, so the coating fixative basically composed of an alcohol base which is doing our work of fixative, and there is carbovax which is also known as polyethylene glycol. Okay, basically it makes a protective layer, thin protective layer, है. These fixative have a practical value in situation where smear has to mailed or sent to a distant cytology laboratory. Basically it is for the transportation purpose, है. And the basic recommendation optimum distance to fix it from about 2 scale, and that is 10 to 12 inches is optimum distance for aerosol fixative. Basically we have to spray from this distance. Prior to staining, slide has to be on capped on overnight in natural wet fixative 95% alcohol for removal of coating fixative. Okay, now there are certain fixative for special purpose, है ना. So special purpose is uh, suppose we have a hemorrhagic fluid or effusion, है ना. There is lot of blood or lesser diagnostic cells, है ना. So we will fix it into the Carnot's fixative, which lyses the red blood cell and make it more relevant or more rich in diagnostic cells. ठीक है, तो sample which are hemorrhagic are fixed by Carnot's fluid. Okay, Carnot's fixative must be prepared fresh Major disadvantage of this fixative are this cause cell shrinkage and nuclear overstaining. Okay, this is about the Carnoy's fixative. And there is second kind of fixative called AAF fixative, acetic acid alcohol formalin fixative. This is ideal fixative for cell block preparation of fluid samples. AAF fixative comprising of 95% ethyl alcohol, glacial acetic acid, and also formalin. Okay, there is certain other kind of sampling technique or smearing technique called imprint cytology. We basically mostly do in lymph node biopsy. There is lymph node, any kind of lymph node, we cut it from half and we just paste it over a slide. Okay, we just touches it, है ना, imprint. Okay, imprint is a touch preparation in which tissue is touched on the slide and it leaves behind the imprint in the form of cells. So basically cell get sticked to the slide. Usually this is the positive charge slide. Okay, which makes better adhesion in between cells and slide. Okay, this is called imprint smear. So earlier I talked about a third kind of sample collection technique which includes the body fluids. Okay, so in fluid cytology we have to preserve fluid specimen prior to processing. Okay, so if you get sputum which has high mucus content, so maximum time it can be kept in 24 hour in a refrigerator. Okay, before processing. Effusion fluids. What is effusion? Effusion is fluid in the visceral cavities, like fluid accumulated in lung cavities or peritoneal cavities. Okay, it has high protein content. Maximum 24-hour is recommended in refrigerator before the processing. Okay, urine and CF, low mucus content and protein content both are low because these are watery. So maximum time can be kept is 1 to 2 hour, usually at the room temperature. Ice is for this thing. Okay, gastric aspirate which has low pH or acidic nature, we have to process it immediately or we can just put it down on ice for some times. Now processing of fluid samples in cytology, we can suppose it is a sputum, so we can make the direct smear or we can make the crushed smear, as I explained in the histopathology class. Okay, moderate amount of effusion if came there is. Okay, so 50 to 100 ml there is, so you just centrifuge it. Okay, and centrifuge it, discard the supernatant, and from the centrifuge you make a smear from the sediment. You fix it in alcohol based fixative or air dried for Romanowski stain or so pap stain, etc. Okay, if fluid is less, for example like CSF or eye fluid, vitreous fluid, okay, then you have to perform a cytocentrifuge. So in cytocentrifugation or cytospin technology or cytosmear technology, you have something this funnel like plastic equipment. Okay, so you just put the sample here and you just cytocentrifuge it. Okay, so you will just the centrifugal force will be pushing the essential cells or content onto the slide. This is positive charge slide. Okay, and you will get this kind of smear, or on realistic way looks like this. This is called cytocentrifugation or cytospin technology, which we will be using for less than 1 ml of CSF or vitreous fluid sample. Okay, this is cytocentrifuge. Now we will, if we will get a large amount of clear fluid, most likely urine. Okay, so we will process it in millivore assembly. Okay, so here is the container, we put lots of urine here. Here is the filter paper. Okay, here is the flask we will collect, which will collect the remnants of urine. Okay, and the diagnostic material will be collected over the, or proteins will be collected over the filter paper, है ना. Now this filter paper will be imprinted on the slide like imprinted smear technology. Okay, it is also positive charge slide which will basically adhere the essential material from the filter paper. Okay, this technology is called millipore filtration or millipore assembly filtration. Okay, if we have to make cell block for immunocytochemistry like we do in immunohistochemistry, the histopathology block, we will make it here a cell block. So in cell block preparation for immunocytochemistry, okay, we will be just basically fixing the specimen prior to processing. Then we will centrifuge the specimen. We will wash with phosphate buffer saline or phosphate buffer solution. Okay, and we will finally discard the supernatant, and now we have the cluster of cells or major diagnostic cells. Okay, so we just add 100 microliter of plasma is human plasma, and we will add the thrombin to it, 30 microliter of thrombin. What is this? In plasma we have fibrinogen, which is soluble plasma protein. Okay, thrombin, what will it do? It will get solid kind of clot, in it will like solid clot, basically a jelly like clot. Okay, and we put this clot on filter paper and process further. Okay, so this is how a cell block is prepared. Now we will be discussing about the stains in cytology or stains of cytology. Okay, there is most common stain is pap stain, most common, which is, and also mgg stain. Okay, pap stain basically get its name from Dr. George Nicholas Papanicolaou, which is also known as father of exfoliative cytology. Now its content of a pap stain. Okay, there is hematoxylin. Harris's hematoxylin is used. This question was asked in exam ki in pap stain which kind of hematoxylin is used. Harris hematoxylin is used. It is basically stains the nuclear part or nucleus part. OG6 for cytoplasmic counter stain. It also stains the keratin component of cytoplasm. Okay. It's a eosin azure EA50 or EA65 is polychrome stain. It consists of 3 dimealy, eosin while light green SF or Bismarck brown. So basic pap stain technique is we do 15-minute fixation by 95% ethyl alcohol. So the rehydration of smear basically means we are using decreased grades of alcohol. Just like we doing 100%, we came to 90% alcohol, we came to 70, we came to 50, we came to distilled water. So tissue is rehydrated. It has water content which will favor the water based dyes. Okay, so nuclear stain we done by hematoxylin dye. Okay, Harris hematoxylin. So after we put it into the hematoxylin, we just it get overstained. Okay, it get overstained. Now we will dip it into a acid alcohol. So this dip in acid alcohol will give it a pinkish color. So to reduce that, we do bluing. Okay, so that's why smear will look like slightest blue type of hue. Okay, weak alkaline solution, Scott's tap water. Scott's tap water we can use. Okay, you just watch the HND video because half of the pap staining is HND. Okay, now after the bluing we are putting the cytoplasmic stain OG6 and eosin azure. Now we do again dehydration, clearing and mounting, and then microscopy. Now in pap smear there are cervical cells. The normal cervical cell, which will be 4 important cell you have to look, because you in some exam you will get question from these cells. So superficial squamous cell abundance show presence of high estrogen in female, will show superficial squamous cell in abundance. Okay, similarly, if progesterone gets high like second and third trimester of pregnancy or in luteal phase, so there will be abundance of intermediate squamous cell. Okay, parabasal squamous cell basically is seen with women with menopause or higher age, which has low estrogen. Okay, basal cell, also known as squamous metaplastic cell, it has highest metabolic activity inside the cell, and here is the you will get abnormalities of cancer or some kind of cervical cancer. Okay, So there are certain reporting systems in pathology. You will get maybe you will get a question, or maybe you will not get. Okay, there is Paris system of reporting, which reports about the urine cytology. So basically, what is reporting system? It standardize the process of reporting of uh, certain sample type. For example, urine or salivary gland cytology. Okay, Million system is for salivary gland cytology. Bethesda system is for breast, thyroid, and cervical pap smear. So cervical pap smear is reported by Bethesda system of reporting. Okay. So pap smear has nuclei which is stained by hematoxylin blue or blackish nuclei. Okay. Cytoplasm non-keratinizing squamous cell blue green and keratinizing squamous cell which is pink and orange, this kind of cell. Okay, now what is specimen adequacy criteria? So basically the terms adequacy means our sample is good for making a diagnosis. Okay, sample is good enough to make a diagnosis. Okay, so preserved squamous cell and endocervical cell, basically the sample we have collected from the cervix of female has preserved the squamous cell and endocervical cell, or and a smear if properly taken should contain the cell from whole transformational zone. Okay, this zone has the highest cancerous diagnostic cells. Highest diagnostic making cell will be present in transformational zone, which is squamo-columnar junction in the cervix. Sample should contain cells from this zone. Okay, and minimum squamous cell will be adequate. In case of LBC, 50,000 cell we should seen on LBC smears, thin prep or Shorpath. Okay, in conventional pap smear there will be 8k to 12k cells should be there. So we must say the sample is adequate if you are meeting this criteria. This is given by Bethesda system of reporting. Okay, now there are other stains, MGG stain, May Grunwald Imsa stain, which has this solution, May Grunwald solution, which has eosin and methylene blue. And there is Jimsa stain. The procedure of stain is very simple, and it is a kind of Romanovsky stain, so it gives the Romanovsky effect. The RBC is look like pinkish, cytoplasm look like blue, and purple will be seen the nuclei. Okay, this is called MGG stain. So let's just take. So let me just take a quick test of you. Okay, the best fixative for routine cytology smear. आंसर आप कमेंट में करिए. ओके, सो लेट मी टेल यू, दिस इज फॉर हिस्टोलॉजी. ओके, दिस इज फॉर cytology. Okay, the best fixative or most common fixative is this 2.5 glutaraldehyde dissociation. It is for electron microscopy. Electron microscopy samples. Okay. Question number 73 basically means CSF is usually processed by which? CSF is usually come to 1 to 2 ml, यानी कि lesser quantity of a fluid. So in fluid cytology we did a cytocentrifugation or cytospin or cytosmear. Okay, this is the technique we use for process the CSF sample. Okay, direct smear we are making for sputum fluid, sputum sample. Millipore is for large quantity of urines. Okay, centrifuge for serous effusion, abnormal fluid collected in pleural cavity or peritoneal cavity. Now we come to the part of waste disposal. So you have to take universal precaution while handling the waste or working in the lab. Universal precaution means to take adequate measure to prevent contact with various body fluids of patient. Universal precaution includes wear mask. You have to wash hand in regular interval. We have to wear gloves. Proper disposal of waste is one of its kind. We have to discuss it in detail because it is in our syllabus. Take care of sharp object. You will get infected if you get cut or punctured by a needle or infected needle or sharp. Okay, wear apron while working in lab or dealing with the waste. Okay, now we will go to the waste disposal. Waste disposal, only one bag or one bin, black bin, is treated by municipality. We just do sanitary in landfill. Okay, but for other kinds of waste, we do specialization technique, just like incineration. It will burn the content of yellow bin at 1200 degrees Celsius temperature. Okay, autoclaving is done for red bean, or dry heat sterilization, also incineration, or for blue bag, also disinfection, autoclaving, or incineration. Okay, black bin includes the general waste, non-inspection general waste like food waste, mineral water bottles, okay, paper waste, etc. Okay, red bin consists of all infected plastic and rubber materials like gloves, tubes with samples, or syringes which has blood contamination. Okay, mostly plastic waste and rubber waste. Okay, here you can read from here: catheters, syringe without needles. Needle will go into sharp container. Okay, uh, in yellow bin, there will be discarding of human anatomical waste or animal anatomical waste like organ, placenta, etc. Okay, dressings or plaster cast, cotton swab which is soiled in blood. Okay, blood bags, discarded blood bags. Okay, which has the positive blood. Okay, reactive blood, HIV positive blood bags, expired blood bags. Okay, expired medicines, etc. This will all go into the yellow bag or yellow bin. Okay, in blue bin we will collect the all broken or contaminated glassware. Okay, these are usually glassware like ampules or broken bottle, etc. Now there is also white container came recently. Okay, which contains all infected sharps weight, just like lancets or needles. Okay, these are sharp objects. Okay, scalpel, etc. So this kind of box is used. Okay, which is puncture proof, leak proof, and tamper proof. Okay, this is the white container. In earlier years we are using only 4. Okay, now we are using 5, including the white container, which separates the metal, basically metal, infected metal waste. Okay, and infected glasswares will go into the blue bin. Okay, so these are the waste management protocol from AIMS New Delhi. Okay, so all the content written here will be a very authentic. So I think this is all about the basics of cytology, and so thank you so much for watching.